Okay, so the last segment of this unit is going to be on mutations. And so this is just a review of what the central dogma says. The central dogma says that DNA can replicate itself, so information can go from DNA to DNA, or the information on DNA can be transcribed into RNA, and then from RNA it can be translated into a protein. And notice where all of these things take place. Replication, of course, takes place in the nucleus, as well as transcription takes place in the nucleus because that's where DNA lives. And then translation is going to take place outside of the nucleus at the ribosome, and that is when RNA becomes a protein. Now, what we're about to talk about is mutations. So where would that happen? Mutations are going to happen where DNA is. A mutation is simply changes in the genetic material of a cell. So that means that mutations would happen here in the nucleus, in the DNA molecule. And then whatever mutation takes place, whatever change or disruption occurs here in DNA, it will play out down here in the protein. Because whatever is changed here, that means the information is different. So the message is going to be different. And so the phenotype will be different. So a mutation is a change in the genetic material of a cell. You can have two different types of mutations. You can have chromosomal mutations, and those are on a larger scale. They're always going to be catastrophic. They're always going to cause disorders or maybe even death. These are your examples of chromosomal mutations. The ones that we are going to spend most of our time talking about will be point mutations, though. These occur in a gene, so it's not going to occur on a large scale with the chromosome. You're going to take a small part of the chromosome, a small part of the DNA, and there will be a change that takes place there, and that would be obviously in a gene. And we call those point mutations because they happen at a particular point in the DNA molecule. You have a type of point mutation is going to be a substitution where one base is replaced with another. And in substitutions, you can have a silent mutation, and that would mean that there's a change in DNA, but it doesn't change the amino acid. Therefore, the protein is not going to change, and therefore the phenotype will not change. Missense means that there is a change in DNA, maybe in one base pair different, there's a substitution with a base pair, but it does result in a different amino acid. This may or may not cause a change in the overall expression of the gene. In other words, this would be kind of like misspelling the word the. Let me use a different example. Misspelling the word cat. C-A-T is the correct way. K-A-T is obviously incorrect, but you can still read it, and it still makes sense. So it's just a little bit different. It's misspelled. But you can still make sense of the sentence. That would kind of be like a missense. Nonsense would be a premature stop codon. Whatever mutation occurs, it causes the translation to stop earlier than normal. And so you get a nonsense protein, something that's non-functional. And then another type of point mutation is going to be a frame shift. And there's only two types of frame shifts. You have insertions and deletions, and most of these, if not all, are going to be catastrophic. The mRNA is read incorrectly because you've taken out a base or you've inserted a base, or you've taken out 3 bases. You could take out a whole codon here or insert a whole codon. So anyway, it's going to produce non-functional proteins, which is going to be pretty devastating as far as phenotypes go. All right, mutagens are just those things that cause mutations to happen. The 3 big mutagens are going to be radiation, like X-rays or UV radiation from the sun, tanning beds, or chemicals like cigarette smoke, nicotine, barbecuing, chemicals that come from the coal being, if you're using a charcoal grill to barbecue. And then infectious agents can be mutagens. Okay, this is a repeat of what I've already said, but there are some pictures here. I think I'm going to go on to the next slide because the picture is bigger. So here is the silent mutation where there's no effect on the phenotype. So here's your DNA molecule, and this is what the normal polypeptide would look like with those amino acids. Here is the silent substitution. Here, there's an A instead of a G. See up here, it's a G, and we put an A there. And so now that's going to change the codon, but it doesn't change the amino acid. The amino acid is still GLY. And that's just because GGU and GGC code for the same amino acid. We can have more than one codon for an amino acid, so that's a silent. A missense here is your normal again, but right here, instead of a C, which is up here in the normal, we're going to replace it with a T. And as a result, that does change the codon to AGC, and it's going to change that amino acid. It may or may not have an effect on the overall phenotype, though. And then nonsense. Here, we're going to substitute an A in for the T. The T is going to be pulled out, and A is going to go in. That's going to change this codon here to UAG, which is a stop codon. This is going to be non-functional. It's nonsense. Okay, sickle cell disease, sickle cell anemia is the result of a point mutation. Here is the normal DNA sequence right here, GAG or CTC and GAG. And then whenever that is transcribed, it becomes GAG. And then that leads you to this amino acid, GLU, glutamine. That's normal hemoglobin. For sickle cell, this A, the T right here, is being swapped out for an A, and that's going to change the codon to GUG and not GAG. And that one change changes this amino acid from GLU to VAL, and as a result, it causes that kind of phenotype change. So instead of having a perfectly round cell, now we've got a sickle-shaped cell, and that's going to cause circulation problems and potentially could be fatal in the person because of the circulation problems that they will have in their blood. Frame shifts, again, this diagram is kind of small, so I'm going to move on to the larger ones. Here is, let's see, we're talking about an insertion here. So here's your normal amino acid sequence right here. And here, what we're doing is if you look up here, it's TAC, TTC. Well, in between the C and the T, we are adding an A, an adenine. And so that means that there is another base pair here. The T will pair up with the adenine. Thymine will pair it with the adenine. And then when transcription takes place, remember you don't have any T's in RNA. You only have U's. You have uracil instead of thymine. So To pair it with this A, you've got to have a U. That makes UAA as the codon, and that's a premature stop. So this is non-functional. That's an insertion. And here is a deletion. So right here, you see up here in the normal DNA, you've got AAA. Well, here that third A is being moved out, is being deleted, and everything from that point on shifts over. And I didn't mention this. That's what a frame shift is whenever you insert a base, that means that everything, the reading frame, you remember how we have to read in sets of 3, the reading frame of codons is going to shift to the right if it's an insertion. If it's a deletion, like what you see here, this A is being removed, so everything past that mutation, that deletion, is going to shift left. And so now you've got a different combination of codons here. In other words, instead of AAA, now you've got AAC and CGA and T. You don't even have 3 at the end here. So clearly this is giving you a different set of amino acids, and this also will be non-functional. Or at the very least, I don't want to say very least, but anyway, at the very least, it will produce a protein that is either it's not functional or it's going to cause a problem. And then this is taking a whole codon out. So this is showing you that it doesn't just have to be one base pair, it can be 3. It can be a whole amino acid missing from the polypeptide. And again, probably going to cause some definite changes in the phenotype. All right. I will be putting up some practice problems for you to look at. We will be going over these tomorrow, so if you don't have time to look at them, that's fine, but if you do, it would make our time together tomorrow much more productive. So try to give it a shot and see if you can build these proteins with these practice problems. And that's it. Thank you. We'll see you tomorrow.